Good afternoon and welcome to the What's on Board briefing from NASA's Kennedy Space Center in Florida. I'm Tabitha Thompson with NASA's Office of Communication. From an instrument to help us better understand cosmic ray particles to research that could lead to innovation in drug therapies, we'll hit a few of the highlights of the dozens of research investigations headed to the International Space Station on tomorrow's SpaceX CRS cargo mission to the International Space Station. First up, for an overview of the research launching, we'll hear from Ken Fields and Pete Hasbrook. Pete is the Associate Program Scientist for the International Space Station Program, and Ken is Director of Operations for CASIS, the Center for the Advancement of Science in Space.
Cool. Thank you, Tabitha.
Thank you, Tabitha.
Thank you all for being here today. It's great to see a packed room, and thanks for all the people who are watching online, watching on NASA TV, and on social media. These briefings are really fun for us to do, and I'm glad to have the opportunity with Ken here to tell you all the cool stuff that we're doing, not only on the SpaceX Dragon launch tomorrow, but I'll also tell you some about what we're doing on the ISS in particular. The Dragon vehicle and the Falcon launcher are very important to us. The Dragon brings us pressurized cargo. It brings us external cargo, what we call unpressurized cargo, but almost maybe more importantly, it's bringing home science samples, and that's a key capability for us to be able to study things in space, bring them home, and then study them more thoroughly on the Earth. Tomorrow's Dragon launch will have on the order of 2,200 kilograms of cargo going up, and that includes that external payload that I talked about. And you'll hear more about that later from the project manager, or he'll introduce what he is really. So we've got over 2 tons of scientific experiments launching tomorrow, and then about 30 days later, I think September 17th is the unberthing date, the Dragon will bring home 1,200 kilograms of scientific cargo for us. That's in addition to the other cargo that it'll be bringing home. The ISS in space is a multidisciplinary laboratory. It's a national laboratory for the U.S. It's an international laboratory made up of the 5 partner agencies, 15 partners participating. At any one time, we've got hundreds of experiments going on. We may have, right now it's probably on the order of 250 different experiments. And out of those, the crew doesn't operate all of those. Many of those are run autonomously, commanded from the ground. Some of those the crew operates, and in some cases the crew is the subject if it's biomedical research. So a very broad spectrum of research that we do. Unlike a lab on the ground that may focus on one type of science and maybe very specifically on a discipline within that type of science, we go across the board, and we categorize our science in 6 different areas. We've got biotechnology and biology, earth and space science, we've got human research, we've got the fundamental physical sciences, we've got technology demonstration, take your hardware to space and see how it operates in microgravity, and finally we do education and outreach. And over the course of the history of the ISS, we'll be at about 17 years continuously inhabited, coming this November. By the way, there's a whole generation out there of young people who have been alive and never known a time when we didn't have astronauts living permanently in space. But over the course of our 17+ years, we've done over 2,300 different experiments on the ISS. That's across the partnership. We've served over 3,300 different scientists who are the lead principal or a co-investigator for research. And we're somewhere around 100 countries that have participated in the ISS over that time. And so our goals for the ISS are to advance the knowledge of our natural world around us. We are paving the way for future space exploration, not just technically in humans, but with the international partnership that we demonstrate as well. We are expanding the commercial market in low Earth orbit. Space station run by the governments isn't going to be there forever. We know there's a great desire for commercial presence in space. We are paving the way not just with commercial rockets and cargo services like tomorrow, but with the companies that are developing hardware and working with our scientists, and Ken's going to tell you a lot more about that. We are really working to improve life on Earth by working off the Earth. Ken's going to tell you more about the science, but I'll tell you some more about what's going on in the space station program. We have extended Peggy Whitson as a crew member on board ISS. She was extended by about 3 months, and that puts her now in the same duration category, almost as long as Scott Kelly and Mikhail Kornienko, who are the 1-year crew members who landed about 1.5 years ago. Peggy was already participating in the fluid shifts experiment, and now by continuing to do that, she is now a new data point in that year-long type of a study. She's also continuing the other biomedical research that she had been doing. After she lands in early September, we've got a new crew launching on a Soyuz in middle September. That will also keep us at 4 USOS crew members. You may know historically we've been at 3 USOS crew members, Americans and the other European, Japanese, and Canadian partners. Now with a situation, a change in the Russian crew planning, we have the opportunity to have 4 crew members. We'd been looking forward to this for a long time with our commercial crew, being able to launch more crew members, but that day is here now. And so with Peggy's leadership and working really hard, and it's hard for us to keep up with Peggy if you've followed Peggy at all, and then coming again in September with the next Soyuz launches for the next year, we'll be at 4 crew. What that does for us is essentially doubles the amount of crew time that we can devote to science. We've already got everything covered, you know, medical, stowage, systems, plus the science we do today, and that crew member's time is going to go distributed totally to science for us, which is a great thing. And so part of the message that we have here is that if you're a researcher and you've ever thought, you know, space is too complicated, Ken will take care of the complications for you, but we now have the time. We want you to come. It's a great time to do the research on space station. Some of the other things going on scientifically around NASA, has everybody heard that there's going to be an eclipse a week from Monday? Good, you guys are plugged in. So the crew, ISS crew, is going to be able to see that eclipse as well. They will actually be able to see it on 3 consecutive passes. Their first pass will be over the Pacific Northwest. They will be able to see the eclipse, the moon in front of the sun. The shadow will not have hit the North American part of the Earth yet. On their second pass over the North America, they'll be up over Canada, but if they look toward the south to the horizon, they'll be able to see the shadow down around Kentucky, Tennessee. And on their third pass, the shadow will have shifted off of the Earth, but they'll see the most full of the eclipse that they'll see. It'll be about 85%. The crew has special filters and special cameras that they'll be using to look at the eclipse. I know a lot of people here want to do that. For tips on observing the eclipse, you can go to the NASA website. It's called eclipse2017.nasa.gov. And finally, the other cool thing that we've arranged for you all, we're hoping for clear skies tonight because there's a great ISS pass over Kennedy Space Center tonight. So about 9:33 p.m., be looking northwest and look for something that looks like an airplane on the horizon. And as you watch it, it'll still come toward you. It'll go right overhead and then disappear just after overhead. It's going into the station's sunset about 200 miles up. If you missed the time, 9:33 local here northwest, or you want to look at it for another city, go to the NASA website called spotthestation.nasa.gov. You can look up any city in the world or any place close to where you want to go. With that, back to the regular program. We're really excited about the Dragon launch tomorrow and all the science capability it's going to bring to us, enable us while it's up there, and then bring home for us. And let me introduce now Ken Shields to take on the National Lab part.
Thank you, Pete, and that was great. What a wealth of information, and it's going to be really tough to follow that act. I got to be honest with you. I mean, it's when you hear Pete run down the list of activities that are going on with the ISS, with NASA, and the international partnerships over the past years, and then see where we are today and what we continue to evolve. It's really mind-blowing when you think about what the future brings. So thank you again. So my name is Ken Shields. I'm with the Center for the Advancement of Science and Space, also known as CASIS. We are the nonprofit, non-governmental organization that manages the National Laboratory on board the ISS. Now, what does it mean, really, to have a national laboratory on board the ISS? Well, really, what it does is it affords almost anyone, essentially anyone, particularly in the United States, with access to utilize the ISS for whatever purpose it is that you deem necessary and appropriate. You could be testing out a technology, advancing maybe some research or discovery in some vaccine development, improving a consumer product. It goes on and on. It's almost infinite as to what you might dream up that you would want to do in the environment of microgravity or the extreme environment of space. So today, in just a few minutes, we have a video where I'm going to let the researchers actually tell you from their voice what they're doing on this mission, SpaceX 12. Every mission we come to, it seems like the portfolio, the projects in the pipeline get more and more diverse and more interesting, and this one's no different. I'd like to take just a few minutes, though, to tell you a little bit about the actual construct of a national laboratory and how that has evolved over years and some of the things that it is enabling. First of all, from a national lab perspective, we represent organizations from a very, very diverse background. We've got researchers from institutions, universities, private sector commercial companies, every industry that you can think of, doing every science discipline that there is. Last but not least, STEM and educational outreach is also a very important mission of the National Laboratory. What the lab has really proven out to be over the years, it's a fantastic example of almost like a research park or a business incubator in space. So on the supply side, not just on the user side, on the supply side, we're enabling private businesses to run their company on the International Space Station. And this launch represents great examples of several companies that are actually supporting some investigations. I'm going to name a few examples, and you'll see some more in the video. One is TechShot. They were here today, and some of you folks may have met TechShot. They're supporting a stem cell investigation on the ISS, but they've already been on the station with a bone densitometer facility. They've done 108 scans on the ISS. This was something that several years ago, I think a lot of experts said you could never do, but it's a facility that's up and running. It's available for use right now. We've got NanoRacks, one of our commercial partners. They've flown hundreds of experiments on the ISS, and they have dozens flying on this flight as well. We have Boeing. I think everyone knows Boeing. Boeing is a fantastic partner for a wonderful STEM education program that we have known as Genes in Space. They're flying their third iteration of Genes in Space on this mission. Space Tango is another private facility on orbit right now. They're flying a second facility actually on SpaceX 13 coming up, also furthering great research. And last but not least, we have a new company and a new facility that's launching on SpaceX 10. It's called the Space Technology and Advanced Research System. They're out of Houston, Texas. The acronym for them is STARS. This is an industry of acronyms, so we have to mention that. So once again, the National Laboratory and this partnership that we have with NASA and CASIS is not only enabling cutting-edge research, it's also evolving this laboratory that we have so that we have the latest and greatest, the cutting-edge facilities that modern-day research can accommodate. And we are also facilitating and enabling business in space and furthering this goal of having a LEO commercial market. So with that, we'll cut to the video right now, and you can learn about more of the research that the National Lab is sponsoring on SpaceX 12.
I'm Greg Johnson at the International Space Station Mission Control Center, and we're here to highlight the incredible research headed to the ISS National Lab aboard the SpaceX Cargo Resupply mission. The Michael J. Fox Foundation will attempt to grow larger, more ordered crystals on the ISS to examine the LRRK2 protein, a main contributor to Parkinson's disease. The Houston Methodist Research Institute will send a stem cell investigation looking at strategies for growing new lung tissue in microgravity. Pharmaceutical giant Eli Lilly and Company will send a freeze-drying experiment to station with the hopes of improving chemical and physical stability within drug products. A new research facility, STARS I, will make its way to the space station to support biotechnology payloads, life science research, and physical science investigations. From Fortune 500 companies to innovative startups to esteemed nonprofit and academic research institutions, this mission exemplifies the wide-ranging opportunities of the ISS National Lab as an innovation platform.
So really, just to sum it up, my take-home message for today's talk, there's never been a better time to go and have access to space and do research in space than what we have today. Pete mentioned it in his talk earlier. We've got unequaled crew time that's on the horizon, right? So we've got our technicians, our astronauts, our people up there who are ready and willing to go and execute and operate your experiments or your technology. We've got unequaled access to the ISS vis-a-vis our commercial resupply contract, and we've got cutting-edge facilities that are coming on like gangbusters from the private sector. So now is the time to go and take advantage of the opportunity to access this awesome platform and accomplish what you need to in your research objectives. Thank you.
Thank you, both Pete and Ken. They're going to stay up here for a couple of minutes to take questions from those of us in the room as well as online. If you're following along with social media, please submit your questions via the hashtag AskNASA. In the room, do we have any questions for either Ken or Pete? The question up front. Bear with just one moment until the microphone gets to you.
We're good?
We're good.
What is it about microgravity that makes it conducive to doing like the biomedical research? Or like, why can't we do that sort of stuff down here on Earth? What is it about doing it on ISS?
Well, maybe Pete and I can tag team it, but if you think about gravity, it's the dominant force here on Earth. It's that you can think of it as that one variable, frankly, in your scientific experiment that you can't control. You can't get a handle on it. So there are things you can do in the absence of gravity or microgravity in ISS and phenomena that you will observe that there's no way you can accomplish on the ground.
So things like growing protein crystals. They will grow larger in microgravity. They'll grow in 3 dimensions. If you grow them bigger and then bring them back, you can study them and start to get a better idea of what that crystal's made up of. Another thing is that you can study biological specimens that have a shorter life cycle, but we still know how they relate to humans. They've been studied for a long time on the ground. We study them in space to see how that changes what we've learned about them in the past.
Do we have another question from the audience? We have a question in the back, and please hold your question until the microphone gets to you.
How often do you have to resupply the ISS with materials?
Let's see. We've got a good rhythm going with our 2 commercial providers. We've got SpaceX and we've got Orbital ATK, and we spread those cargo vehicles really kind of where we need them. So we work them around things like the Soyuz launch and landing, where we're rotating crew members. We work them around things like thermal constraints and spacewalks, like we have coming later this week. But I think it's on the order of probably 6 or 8 flights per year, spread among the 2. And both of them bring a lot of cargo to the space station. SpaceX is the one that brings the return cargo home for us. So again, we kind of alternate between the 2 of them. It gives us redundancy and flexibility as well.
We have time for one more question from the audience. Please hold your question until the microphone reaches you.
It's kind of a more general question, but I'm curious about the earth science projects that have gone up. Do you recall what some of the projects were, or were they regarding? I'm curious just about what an earth science project would be like at the ISS.
I can give you a couple examples, and I'm sure Pete has several also. National Lab, over about the past year and a half, we've sponsored an investigation that's called Tropical Cyclone. And Tropical Cyclone is a team out of Cambridge or Boston area. They are using crew Earth observation imagery and looking at the cloud tops of cyclones or hurricanes and applying different algorithms to learn how to better forecast, particularly in very remote areas of the planet where they don't have all of the forecasting facilities that we have in the United States. There's one good example. Another example, we have once again, a private facility on board station called Muses, which is a facility class payload that can host at least 4 or maybe more all types of remote sensing Earth imaging payloads or projects that could serve a number of Earth science disciplines.
So I had a couple others that came to my mind. One of them is called HICO, or the Hyperspectral Imager for Coastal Oceans. When you take a picture here, you're essentially getting probably red, green, and blue pixels. A hyperspectral, or many of the spectra, takes up to 100 different bands. So you get, for every single pixel you see, you get 100 colors. And we had that as mounted on the exterior of ISS. Had another one that was called ISS Rapid Scat, or Rapid Scatterometer. And it basically shot a laser down at the ocean, and based on the response, the reflectance could tell which way the waves were going and what direction was the wind and how fast was the wind going on the ground. And the other neat thing about that is, as it was taking data, the whole data transfer system, downlink and distribution, got it out to people like NOAA and weather forecasts within about 2 to 3 hours of the ISS going overhead. So,
Thank you to both Ken and
Thank
to
you.
Pete.
And good luck tomorrow, everybody.
Pete talked for just a moment about the spotthestation.nasa.gov website. Not only is that a great way to track the space station, but it's also a great way to impress your friends if you know when the space station is going overhead. The other thing I like to do when I see the Spot the Station app or the notification is think about who's flying overhead. So it's always good to look at the crew launching at that point, or on board the space station at that point and just know who's in the space station as it flies overhead. The other thing, Ken mentioned protein crystallization as one of the benefits of microgravity. Our next speakers could talk to you at great length about that. We have Marco Baptista, who's Director of Research Programs for the Michael J. Fox Foundation, and Sebastian Mathia, who's Lead Investigator for the crystallization of LARC-2 under microgravity conditions. Marco?
Yeah, thank you so much. So my name is Marco Baptista from the Michael J. Fox Foundation, and but just before I talk a little bit about Parkinson's disease and why we're doing this experiment on the International Space Station, we have a brief video that we'd like to show you.
I want to thank the Center for the Advancement of Science and Space for helping our mission grow beyond the boundaries of Earth's gravitational pull.
As one of the most common genetic contributors to Parkinson's discovered to date, LARC-2 is the focus of intense investigation by Parkinsons researchers.
So LARC-2 is one of the most common genetic causes of the disease, and for over 10 years, no one has ever been able to crystallize and solve the structure of LARC-2. And we're hoping that under microgravity conditions, we will improve the resolution.
It has been proven in the past decades that crystallization works better under microgravity conditions, and we hope that this is true for LARC-2 as well. Having the atomic structure will allow us to design LARC-2 inhibitors, and those LARC-2 inhibitors can be developed into drugs in the end.
We're incredibly excited to be able to work with CASIS on this project, and we're willing to go to the ends of the Earth to try to solve the crystal structure of LARC-2.
We're thrilled that PD Research has been selected to travel to the International Space Station, and honored to partner with CASIS on behalf of the PD community here on Earth. Thank you.
Great. So I just want to make some brief remarks about Parkinson's disease, and then we'll switch it over to Sebastian. Will give you a little more of the details of why we're doing this experiment. Parkinson's disease is a progressive, chronic neurodegenerative disorder which affects about 1 in 100 people over the age of 60. The estimates vary, but we're thinking there are about 1 million people in the United States that have Parkinson's disease, and possibly over 5 million people in the world. And the disease is characterized by both motor symptoms, such as rigidity, a slowness of movement, an abnormal gait, and also non-motor symptoms, such as cognitive deficits, autonomic dysfunction, and depression. And there are treatments that ameliorate these symptoms. However, we still do not have anything that is disease-modifying that will either reverse, will stop, or will slow down the progression of the disease. And that's why at the Michael J. Fox Foundation, we're very interested in this particular gene called leucine-rich repeat kinase 2, or LARC-2, as we like to call it. And the reason that we're so interested in it, and that we've invested already over $100 million into this potential drug target, are for the following reasons. So first, mutations in that gene have been shown to be one of the most common genetic causes of the disease. Second, the clinical manifestations of the disease in LARC-2 carriers is actually very similar to the non-genetic, more common forms of Parkinson's disease. So the possibility that a therapy that treats LARC-2 mutation carriers could actually possibly treat everybody who has Parkinson's disease. Also, it's very druggable. And that term is used when the pharmaceutical industry has a lot of expertise in being able to target that particular protein. So there are a class of inhibitors called kinase inhibitors, and the pharmaceutical industry is very good at making those classes of drugs. So it's low-hanging fruit to go after LARC-2. And finally, there's converging biology that shows that LARC-2 is playing a very important, if not a central role in Parkinson's disease. So we've made quite a bit of breakthroughs recently on LARC-2. We've been able to identify its substrates. A substrate is a protein that is either being turned on or turned off by LARC-2. We're also doing quite a bit of work on preclinical safety, which is showing us that it's possible that we're going to be able to move and do clinical development of these LARC-2 kinase inhibitors in people. But we still need more breakthroughs. And one thing that we still don't fully understand is what LARC-2 exactly looks like, what its atomic structure is. And we have a world-class team that's working on this problem here on Earth with a lot of different techniques. But we have one particular technique that it appears that we possibly might have better luck if we were to do it on the International Space Station under microgravity conditions. And this is looking at X-ray crystallography. And the basis behind that is that you're able to better understand what a protein could look like, and therefore, with understanding its structure, you could better understand its function. So you want to take the protein, make it into a crystal that amplifies the signal, amplifies the protein. You bombard it with X-rays, and then you're able to better understand what the protein actually looks like. This will help in two ways. First, it will help you better understand the biology of LRRK2. If you understand structure, you can understand function. And secondly, although the pharmaceutical industry has been pretty good at making LRRK2 kinase inhibitors, they've been doing it pretty much blind in the sense that they're using other kinases, other proteins that have been solved, and using that to make assumptions of what LRRK2 looks like and making drugs against it. And they're doing a good job with it, but we'll never know if we can have the most optimal LRRK2 kinase inhibitor without a high-resolution atomic structure of LRRK2. And so I'd like to pass this on to Sebastian, who can explain to you a little bit more about the experiment on the ISS. Thank you.
Yeah, thank you very much, Marco. So I am part of the team that is applying the technique X-ray crystallography in order to solve the three-dimensional structure of LRRK2 protein. And our team is based in Oxford, UK, and in Frankfurt in Germany. Our general workflow is like, first we make a lot of the protein, then we produce crystals of this protein, then we analyze those crystals using focused X-ray beams, and finally we calculate the three-dimensional structure of the collected data. Yeah, and our experiment on board the ISS, our aim there is to generate high-quality crystals. LRRK2 protein is huge. It's a massive protein, 2,500 amino acids. It comprises 7 domains. This is a lot for a single polypeptide chain. We believe that LRRK2 is capable of adopting a multitude of conformations, and in addition to this, LRRK2 can oligomerize. All this makes it a rather challenging target to work on. We managed to produce a shorter version of the protein that is better behaved than the full-length protein. Yeah, and this protein is crystallized then. For crystallization, we use this tool. It's a chip manufactured by the company Anatrace. In each chip, there are 16 tiny channels. We fill the first half of each channel with the LRRK2 protein. Then we fill the second half of each channel with an additive solution. And after starting the experiment, the protein diffuses into the additive solution, and the components of the additive solution diffuse into the protein solution, and eventually at some point the protein crystallizes. The experiment is like, so we set up the channels on Earth and flash freeze them immediately after setting up, and this is how they are transported to ISS. This will happen hopefully tomorrow. And the astronaut then has to place our samples at ambient temperature. This is when crystallization is going to occur. And after approximately 4 weeks, the samples are transported back to Earth, and this is where we analyze the crystals and where we hopefully are going to solve the structure. Yeah, we did this quite often on Earth, and we managed to make crystals reliably. So the experiment is stable. We can make as many crystals as we like. So why do we send the samples to space then? Could you please do the next slide? This is simply because the quality of our crystals is not high enough. Crystals are just not good enough if we make them on Earth. You see it there that the edges are not really sharp. You see that the crystals are a bit streaky. And one reason for this might be that on Earth we have gravity, And yeah, you asked the question before, on Earth we have gravity, and that means that more dense particles will sink to the bottom of our sample, forming unwanted gradients. And another thing is that subtle changes in temperature will lead to convection. And this is what we don't want. This is bad for crystallization, and we hope to overcome this if we do the crystallization on board of the ISS. Because there are microgravity conditions for the sample, this feels as if there was no gravity, and we don't have any unwanted gradients, and we don't have convection there. And we expect this to result in slower crystal growth and bigger crystals, more regular crystals, more perfect crystals, and this will ultimately lead to higher data quality. If we get a structure of LRRK2 protein at 6 angstrom resolution, we will see the domain architecture. We will start to see secondary structure elements, and this will help us to understand the mechanism of action of the protein. We will see how it is regulated, yeah, and we will understand Parkinson's disease better. And if we get a data set with a resolution of better than 3 angstrom, then we have atomic resolution, and with this data we can do rational drug design, something that Marco has mentioned already. So I say it again, at 6 angstrom we see the domain architecture, we understand Parkinson's disease better, and at 3 angstrom we have atomic resolution. And our Earth-grown crystals, they are at 7.5 angstroms now. So we are not far away from the 6 angstroms. And yeah, we have justified hope that we will get some interesting results from this. Thank you very much.
Thank you to both Dr. Baptista and Dr. Mathieu. They're going to stay and take a couple of your questions. So if you have questions in the room, please raise your hand, wait for the microphone to get to you. And if you have questions and you're following along online, please submit your questions via the hashtag AskNASA. First question in the room on the front row.
Hi, my name is Sarah from Futurism. You mentioned the mutations in LRRK2 are what kind of contribute to the disease. So as we better begin to understand the structure of the protein, could we potentially use things like CRISPR to edit out these mutations as opposed to using the inhibitors?
Yeah, that's a great question. So right now we utilize CRISPR-Cas9, which is a technique that really makes it a lot easier to do gene editing in a lot of the research that we do. As a particular therapy, most drug companies, and what I was mentioning before, where I think we have the best shot at trying to get a therapy, is not to do the gene therapy, but is to do the kinase inhibitor therapeutic approach. And I mentioned before, it's because there's so much expertise in doing that. There's past experience within the oncology field that this is an approach that could work. And we think that's our best shot. But we're definitely keeping an eye on CRISPR-Cas9, and we do use it quite a bit for our research.
And we do have a question from social media, so we'll start here.
Hi, yep, I'm Emily from NASA Social Media. This question comes from Twitter. Are the proteins tested extracted from a particular cell or product?
Yeah, the proteins are human proteins. That means the sequence is the same as the one for human proteins, but we make those proteins in insect cells with the baculovirus system. And that's how we can make a lot of the protein. Extracting it from human source will not be sufficient.
Okay, thank you. Do we have any other questions in the room? We have one question toward the back. Please wait for the microphone to reach you.
I'm just curious, with the results that you guys are going to end up with, hopefully some positive stuff, are there other disease organizations that are going to benefit from this, that are kind of watching you guys to see what your results are?
So in case we will get the structure, we will of course publish it as soon as possible and make it available for the public.
Yeah, definitely everyone in the Parkinson's field looks at other neurodegeneration fields. We monitor what's going on in Alzheimer's. I think they monitor what we're doing also. At the highest level, that's the type of monitoring that we do to see where the similarities are and where the differences is. Specifically with this experiment, I'm not sure how this will be affecting other disease indications, but as Sebastian said, we try to get the information out and people can freely access and see what we found.
Thank you both for your time. Thank you. Our next highlight is the effect of microgravity on stem cell recellularization, also known as the lung tissue experiment. For that, we have a video ready for you.
We've created a model of the human lung that has most of the cells in it that a lung would have, and we're going to use this model to evaluate what happens to your lung on long-term spaceflight. We're using stem and progenitor cells. They're the cells that give rise to the cells that allow you to have gas exchange and breathe. The larger mission is to eventually someday be able to, instead of make pieces this big, is to make a lung this big for transplantation for somebody. If you're going to look at what atoms do, that affects a molecule, that alters a cell, that changes how a tissue responds, that alters how an organ functions, that changes how you function, you have to be able to walk it back and say, I can look at it at these small levels and build this picture of what's happening in a person. That all may be very different in space because microgravity influences everything that a cell does. And the opportunity to have a chance to send an experiment to space and get data back is huge for a scientist. It happens once in your lifetime.
Unfortunately, Dr. Nichols was unable to be with us today, but we do have Ken Shields, we'll bring him back up from CASES, who to answer a few questions about the lung tissue investigation.
Questions are not required.
Do we have any questions in the room? Do we have any questions on social media? Once again, you can submit your questions using the hashtag AskNASA. Any questions?
You're welcome.
Thank you. Our next investigation is the cosmic ray energetics and mass investigation. We heard earlier in the pre-launch news conference that the astronauts will be getting ice cream on this delivery. They'll also be getting ISS cream. Sorry. Jacob Smith from the University of Maryland.
Thank you. Yes, the name of my experiment is Cosmic Ray Energetics and Mass for the International Space Station, and so we've nicknamed it Ice Cream. The goal of the ice cream experiment is to extend the energy reach of direct measurements of cosmic rays to the highest energies possible so that we can study the cosmic ray origins, where they come from, their acceleration mechanisms, how they got the energies that they have, and how they propagate, what's their history through the galaxy, and how they interact with the stuff that's between the solar systems. So I'd like to tell you about the instrument a little bit. We have a video, if we could start that. You can see what the instrument looks like. There it is rotating, ice cream. It's about the size of a large refrigerator. And the top piece is the grapple fixture that the robot arms use to move it around. It will be located on the JAXA Experimental Module Exposed Facility. There you can see the inside. On the right is the space where the electronics go, and on the bigger area, that's where all the detectors go. We stack them up so we can maximize the field of view. At the bottom, we have detectors that help us to separate electrons from protons. We measure hydrogen, helium, all the way up to iron, basically the elements of the periodic table. We have a calorimeter to measure the energy. It's made of tungsten. That's surrounded by detectors used to trigger when cosmic rays pass through. We have a target so that these high-energy hydrogen atoms can disintegrate, and we can measure the energy in the calorimeter. We also measure the charge with a silicon charge detector at the top. So in other words, it's a counting experiment. So simply by counting cosmic rays, you can tell where they came from, how they were accelerated, and what happened to them along the way. So one of the benefits for being on the space station, there's two reasons. One is due to the altitude, and the other is due to the duration. So CREAM started out as a balloon experiment. We flew 7 flights over Antarctica for a total of 191 days over those 7 flights. And that altitude is about 40 kilometers. But on the space station, we're at 400 kilometers. So being at that altitude, you don't have to worry about the atmosphere. The problems that the atmosphere has with direct measurements of cosmic rays is that they can interfere with how we identify the charge. And the identification of the charge helps us to understand the propagation mechanisms, because some of the cosmic rays, such as boron, are produced through interactions of carbon in the interstellar medium. That's the stuff that's between the solar systems and our galaxy. Yes, so the other reason is due to the time duration. So a single balloon flight lasts about 30 days, but on the space station we have a goal of 3 years, but we plan to be on for the life of the station. So going from 30 days to 3 years, or to the life of the station, that extends our exposure time by an order of magnitude. And by doing this, we improve the statistics so that the uncertainties that are that come out of the measurements are reduced. We maximize the energy reach, because cosmic rays, there's less of them at higher energies, exponentially less. So you really need to stay up for a very long time so that you can measure the higher energy particles. And so by having larger statistics, being able to measure higher energies, we can use this to distinguish various models in the theory. So Ice Cream is not the only cosmic ray experiment in space. There's many others. Just a few examples are Pamela, AMS, which is on the space station, Calette, Dampy. These are also direct cosmic ray experiments, but they measure at a little lower energy. However, their high energy has some overlap with our low energy, so the data that will come from Ice Cream will not only complement their measurements, but also add to them. There's also indirect measurements of cosmic rays. So you can measure cosmic rays after they interact in the atmosphere and they break up. So these experiments allow you to reach higher energies, but you can't tell the source as well. So our high energy reach overlaps with their low energy reach. So with all of these instruments and all these projects measuring cosmic rays, you can develop a fuller understanding of the cosmic rays in our galaxy. And I think that's it, yeah.
Okay. Thank you, Dr. Smith. And if you'll stay for just a moment for a couple more questions. Do we have any questions in the audience? If you do have a question, please raise your hand and wait for the microphone to get to you.
Hi. What do you need the information for? Is it just for understanding them and knowing more about it, or is it to measure how it affects human biology?
Well, cosmic ray physics for us is a fundamental science. So at this point, we've identified what they are, that they come from outer space, but we want to pinpoint the sources. Where do they come from? How do these objects in the universe create them? What kind of mechanisms can accelerate them to the energies that they have? The energy range that we measure them are higher than what we can make on Earth. So we want to understand these processes. And yeah, so it's a fundamental science. The energies are too high to affect the ones that we measure, biology and space science, but there's lower energy ones that do have these other effects.
Do we have any other questions in the room? Thank you, Dr. Smith.
Thank you.
Our next speaker is Michael Delp, Principal Investigator for the Rodent Research 9 investigation from Florida State University in Tallahassee.
Good afternoon. It's good to be here with you. So I'm part of the Rodent Research, and this is a program that's sponsored by NASA Space Biology. And really it fundamentally looks at some of the biomedical problems that occur in humans with space flight. And we use rodents to try and better understand some of those problems, and then really with better understanding, to develop or propose more effective countermeasures of what we can do in space and even after space flight to counteract that. So the mission that I'm part of is Rodent Research 9. We're looking at two different biomedical issues. The first is visual impairment that occurs in some of the astronauts. To date, it only occurs in male astronauts. And so we're looking at a couple of different aspects of how visual impairment may occur. And the first thing we're looking at is the vasculature and the pressure in the eye. And if I can have the first slide, please. You can see in the eye that there's a pretty dense vascular network. And this vascular network controls something called the blood-retinal barrier. Now, many of you have heard of the blood-brain barrier, which is a very tight control of fluids and proteins and other molecules that cross into the eye or into the brain. And so this is the picture of the blood-retinal barrier. We'll also be looking at the blood-brain barrier in the brain and looking at different ways that fluids pass from the vasculature into the extravascular space and how that can affect either intracranial pressure or interretinal pressure. The second thing that we'll be doing is really looking at the brain circulation and how that affects blood pressure within the skull. And as part of the circulation, there are three aspects of fluid moving in and out of the brain that could affect this. One are the arteries and the blood going into the brain or skull cavity, and two, the veins that bring blood and fluid out. And a lesser known vascular system that's in the neck that drains the head and the brain is the lymphatic system. And I have a short video that shows some of the action of the lymphatic vessels that we isolate and study. So this is an isolated lymphatic vessel. And when you think about the pumping that occurs in the cardiovascular system, you usually think about the heart. The heart has a rhythmic pumping. It has valves that keep blood or fluid moving in one direction. Well, if you see in this, in the very middle of the picture, you'll see a set of small valves. And those valves keep fluid moving one way toward the heart. And these lymphatic vessels are throughout the body, but it really is the lymphatics in the neck that helps determine the blood pressure, or the fluid pressure, in the brain as well in the eyes. And so we're going to be looking at this. We're working with several other scientists on this interocular and intracranial pressure. Dr. Vivian Mao at Loma Linda University will be studying the eyes with us, and Dr. David Zaveja from Texas A&M University will be looking at the lymphatics, and of course my lab will be studying the arteries and the veins. The other biomedical issue that is being studied with these animals is locomotion. Because microgravity, you have a fairly severe physical inactivity that can affect a number of organ systems, such as muscle and bone loss that you've heard about. Well, we're in this project, it's really Dr. Jeff Willey from Wake Forest College of Medicine, and he'll be looking at how the animals locomote before they go into space, and then once they get back, how their locomotion has changed. And then specifically, he's also going to be looking at the cartilage in the joints, because with severe inactivity, you get degradation of the cartilage in the limb joints, and so he will be studying that in these mice. So really, again, rodent research is geared toward helping us maintain and understand health issues with astronauts, particularly as it relates to long-term space flight and being in space for months to years. So thank you.
Thank you, Dr. Delp. And Dr. Delp will stay while you take a few questions. Do we have any questions in the room? And please do hold up your hand and wait for the microphone.
Thank you. What makes rodents the ideal candidate for this type of research as opposed to other animals?
Well, there are better animals. For example, non-human primates would be probably the ideal. But with rodents, both mice and rats, you can fly larger numbers of the animals. The other thing is that rodents and mice and rats, they have their lifespan is shorter. They have a higher metabolic rate. So changes that occur in weeks in mice and rats take months to do. So 30 days of a mouse being in space is roughly the equivalent of 3 years of a human. So we can really get a very good look at what long-duration spaceflight will be like, particularly as we think about manned missions to Moon and Mars.
I believe we have another question in the room. Please wait for the microphone.
Yeah, Jose Andrade from Engadget.com. Do you expect to have several different generations of mice in there, or is it just one generation that you're sending?
Just one. It's 20 mice that we'll be sending. Yeah. They'll be going up at 6 weeks of age. Well, unless there are delays.
Do we have any other questions in the room? Please wait for the microphone.
Is the visual impairment temporary or permanent?
Well, there were some changes in visual acuity that was seen in the shuttle astronauts, but it wasn't until the astronauts were in space 4 months and longer that there started to become visual changes that were permanent. And so with the permanent changes in vision, that's when you become more concerned. And in 2010, NASA, held a conference to really bring out this problem, to bring experts together to try and then look at what might be the underlying causes for this. And so this is really a continuation of that effort.
And we do have one question from social media.
Yep. This question comes from Alexis Reyes on Twitter. Do mice mind being in space? And also, how is this research going to be applied to other aspects of gravity effects on the body?
So do the mice mind being in space? Well, it's hard to know exactly what a happy mouse looks like, but I will say this. One of the things that was amazing to me was that the mice, when they're up in space in the cage configuration that they actually use the cage as a running wheel. And they will run around the cage. So you might think they would just float around there, but they learn after a couple of days, and one of them will start doing it, and then several, and before you know it, the whole cage. And then if one cage is doing, there's an adjacent cage, and a day or so later, the adjacent cage will start doing. And so there have been studies that if you put a running wheel out in the middle of a field, wild mice will come on and get on that running wheel and start running. They don't have to. They have exercise for other reasons. So I would say that there are some indications that they deal with, and it's a stressful environment. I mean, it's stressful for humans, and so they have ways that show that they cope with it apparently pretty well.
Thank you. Thank you. Any other questions in the room? Okay. We do have one more question. Please wait for the microphone.
Yes, might this research help identify other sorts of eye diseases that people suffer from here on Earth?
It's possible, any time that you are able to elucidate factors, say, that can affect vision in one way, it's a possibility that it can have the same effects in other conditions in ways that you didn't think about. So I think even if it's not directly applicable, the biology of understanding the various factors that can play a role in visual acuity and how that would change, I believe helps us better understand our organism as a whole and then how it adapts to situations of different environmental conditions, and gravity being one of those conditions.
Thank you, Dr. Delp.
Thank you very much.
Just a reminder for those who are following along, either on social media or in the room, you can use the hashtag AskNASA to comment on the briefing or ask a question. Our next presenters are John London, Chief Engineer for the U.S. Army Space and Missile Defense Command, and Wheeler Chip Hardy, who is the Kestrel I Program Manager for the U.S. Army Space and Missile Defense Command. Please join us.
Thank you, Tabitha, and good afternoon, everybody. We are very happy to be here. We are going to talk to you about a program called Kestrel I. Kestrel I is a satellite. Unlike the other research and technology you've heard about today, Kestrel I is a free-flying satellite that will be deployed from the space station. It's a program that we've been working on for about 5 years, this particular satellite. If I could have the first slide, please. You can see a picture of it there. It's a little hard to tell how big it is. It's about the size of a dorm room refrigerator. And compared to many satellites that fly in space today, that's relatively small. But it's a testimony of the technology, the revolution in electronics technology that's taken place in the last 20 years. Certainly, there's no better example of that than holding the smartphone in your hand and realizing the computational power that it has. But we have extended that paradigm into space, and we're not the only ones. There are a lot of people that are doing it, both in the government as well as in the commercial sector, that are taking advantage of this incredible power that you can pack in a very small space. This particular satellite is designed as an imaging satellite, and it's important for the Army to be able to do imagery. And what sets this apart from other systems that have been developed in the past is it has the capability to directly send that image to the user on the ground, and to do that in a very short period of time. From the time it's asked to take the picture to the time it sends the picture down to the ground and appears on a laptop on the ground, very, very short period of time, measured in maybe 1 to 2 minutes. So I'm going to turn it over to my colleague Chip Hardy here in just a moment. Let me have one more slide, please, and I'll turn it over to Chip. Have the next slide, please. So that this really tells you the 3 key tenets of the program. The first one is low cost. It is not only small, but it doesn't cost much. And that's important for anybody that's developing space systems, but it really is remarkable how low cost these systems are today and still have the capability they have. The second point is survivability. They are inherently survivable, operating in low Earth orbit. And finally, and most importantly, the one I've already talked to you about is the responsiveness aspect, the ability to get that imagery to the ground very, very rapidly. So I'm going to turn it over to Chip and let him talk to you about the specifics of the spacecraft. So Chip, take it away.
Thank you, John. As John said, my name is Chip Hardy, and I'm the Government Program Manager for the Kestrel I development effort. As John indicated, Kestrel I is a small microsatellite class satellite whose job is to provide tactically useful images to the at the tactical level to the user on the ground. What we are doing off the space station is an experiment that, if successful, could lead to an objective system. The main main benefit of Kestrel I is not to provide high quality images, but rather, being low cost, you can have many satellites to populate a constellation in order to have one persistently or frequently overhead to provide some persistent presence. And then the final point to make is that being low cost and having many satellites, when you launch or having a failure at launch or an anomaly with a single satellite unit does not lead to a loss of service. Therefore, you have graceful degradation. It is a Joint Capability Technology Demonstration Program. This is a special program within Office of the Secretary of Defense. And as such, we have a COCOM sponsor, Combatant Command sponsor, the Pacific Command, once we're on orbit, a large part of the experiment will be to evaluate its utility, and so the PACOM will do an assessment of that.
Chip gets the tough duty of doing the experiment over in Oahu, so he'll have to spend at least a month over there. But somebody had to do it, and Chip volunteered.
That's right. So I think subject to questions, Tabitha, that's all we had.
Okay, great. Thank you both. If we have questions in the room, please raise your hand. Wait for the microphone to make its way to you. And if you have questions and you're following along online, please submit that via the hashtag #AskNASA. Any questions in the room for these gentlemen? We have a question up front. Please wait for the microphone.
What kind of devices will be on the ground receiving images from this satellite?
So we have, for the JCTD, like I said, in the experiment, we have 2 ground stations. One is at our place of business, Redstone Arsenal, Alabama, and the other one will be at Pacific Command in Honolulu. The one at Redstone is a fixed site antenna. The one at Pacific Command is a highly mobile antenna called the Gator, and it is an inflatable antenna that has great logistics impact in that it is lightweight compared to a traditional antenna. You save about an order of magnitude in weight and still get the same performance.
The antenna that Chip's talking about, if I can just add to that, it is very lightweight, and it can be, you can take the entire antenna system, it's a 2.4 meter parabolic dish antenna, and you can, and it's inflatable, so you can actually pack it on checked baggage on a normal commercial airliner and take it wherever you need to go. So it's really great from that standpoint. And just to underscore what Chip's saying, the ground station consists of the antenna and a transmit and receive radio and a laptop. And that's it.
We have one question from social media, and then we'll take the next question in the room.
Yeah, this question comes from Twitter, and it asks, could you tell us about any civilian or emergency uses for for response to national disasters?
First of all, one of the key roles that the military has today is in humanitarian assistance. You know, when there's a hurricane disaster, for example, in the Caribbean, the United States military would be called to help and assist in that. Oftentimes, communications and imagery information are lacking in those kind of situations. This could provide tremendous capability for that. It certainly could be available for other government agencies, including civil agencies like NASA. So it has multiple applications potentially.
And we have another question in the room.
Hey, Lisa Gonzalez with Vice. I had a similar question, but once you have this information and this intel and these images, say of a natural disaster, what are you looking for? How does that information help you, help the Army, help anyone make change or do something good?
Well, one aspect would be if you had an objective system, Kestrel I, and had a constellation, then you can monitor the situation and provide changes to the people trying to address it more rapidly.
Right. I mean, you know, you've probably heard of these commercial applications for imagery where they'll monitor a parking lot in a commercial company and use the progression of the parking lot over a period of days, months, and even years to determine, is that business growing, is it shrinking? So there's lots of applications like that, but just from a military standpoint, certainly trying to understand what the condition is in an area where the Army might be operating and be able to know frequently. The key really is having rapid refresh of the information so that it's not stale.
Any other questions in the room? Okay. All right, thank you, gentlemen.
Thank you.
And last but certainly not least, we have a group here today to talk to us from the Boy Scouts of America. We have Elliot Lee, who is a scout with Troop 209, Andrew Frank, Scout with Troop 209, and Norman McFarland, the team leader, also with the Boy Scouts of America.
Hello, my name is Elliot Lee, and first slide, please. I'm going to be talking to you about the experiment, and I'm going to introduce you into what we're about to do. Oh, can we have the first slide? Yeah. So our experiment is we're going to use an organism and a known cancer-causing substance, and we're going to use that to measure the time and mutation rate on the microgravity versus gravity on Earth. This could be actually a huge impact on human tissue growth / cancer research. The Ames test is originally used with an organism and a suspected cancer-causing substance, and but we use the direct opposite of that, and we use a known cancer-causing substance and an organism to measure the rate between Earth's gravity and space gravity. Next slide, please. So as you can see, we have different concentrations, and each concentration has different mutation rates. Also, some of the Ames test, it could originally be used, and some of the concentrations that it uses is 4-NQO, also known as nitroquinone oxide. Oh yeah, what was it? Oh yeah, and our not an accountant. Yeah, we use our concentrations are 1 to 2, 1 to 4, 1 to 8, and so on until 1 to 64. And some of our original well designs were we were going to use actually an entire well plate, but that was too hard, and we were going to make it circular, but that was also too hard. So we made an octagon-shaped cylindrical use, and we made it 5x14, 8 well plates. And next slide, please. Yeah, so in this slide, after the test, we're going to be reviewing the pictures, and once we review the pictures, we're going to log them by hours, and we're going to count how many mutation cells have happened. But we have to take note of the environmental effect around it. So here are the 3 sensors: temperature, humidity, pressure sensor. Then there's the light sensor, and then there's the radiation sensor. We got to take note of each environmental effect to see how each affects the mutation rate, and they're going to be logged into our SD card in the NSC board. Once these are logged, we're going to test our hypothesis, and our hypothesis is, will the E. coli mutate faster in microgravity than on Earth's gravity? And I'll pass it on to Andrew Frank.
So basically, we have all these sensors, and we're trying to get the environmental data back so that we can run a similar test on Earth as close as possible to the one that's running on the space station, except for gravity. And so that's why we have all these sensors. We actually had so many that we had to use an integrated communication system because we didn't have enough inputs on our NESI board, which was provided by Texas A&M. Next slide, please. So the organism we're using, we froze it in -20 degrees Celsius freezer, and that's to ensure that the experiment doesn't begin before it actually gets to the space station. So here you can see that's our NESI board. We were soldering wires directly onto it, and it was causing a lot of damage. So if you go to the next slide, you can see we have a custom circuit board that holds 2 cameras and the sensors to monitor environmental variables within the chamber, as you can see here. If you go to the next slide, You can also see this other custom board that we've created, and that whole that connects everything. It really cleaned up a lot of the wires for us. If you go to the next slide, you can see this is a sketch. We tested a bunch of motors to find the perfect one that worked with this test. And this is just a sketch of one of the assemblies we used for testing it. If you go to the next slide, you can see that we are not the only people working on this. There's like a whole team behind it. You can see us. We took a picture with Mark Polinsky, who was an astronaut on the ISS. And if you go to the next and final slide, you can see all our sponsors that we would like to thank for helping us out. And now I'll pass it off to our adult leader, Mr. Norman McFarland.
Hello, everyone. As project leader, I could not be more proud of these boys. I could also use the microphone. As project leader, I couldn't be more proud of these boys. They started off as a bunch of bright-eyed school students, and now they're self-starting, problem-solving young engineers. They're just doing they've done a phenomenal job. For those of you that think that scouting is all about camping or campfires or knot tying or whatever else your preconceived notion, maybe you can see now that we do some STEM stuff once in a while. We actually offer over 60 merit badges in the science, technology, engineering, math area. In addition to that, we have several different Novus awards for special studies into the STEM area, and beyond that, once you have some NOVA awards, you can earn Supernova awards, the top of which belongs on your college resume as a significant event there. So our team would like to thank NASA for this TV time, CASIS, NanoRacks, Texas A&M University for sponsoring this team, General Kinetics, EBPI was our biology source, and Spark Fund for many of the sensors that you saw here. Finally, we have a very special thanks going out to two people, Nan Elder and Lisa Early, who were former Scout Council and district STEM professionals that set up this program so that we could be up here today. We also, of course, would like to thank our sponsoring organization, the First United Methodist Church of Palatine, Illinois. Thank you for your time.
Thank you all for your presentation. Do we have any questions in the audience for the Boy Scouts? Do we have any questions on social media? Oh, we do have one question. Wait for the microphone, please.
I'm just curious, how long has it taken you guys to get from the idea to do this to where you are now?
It's been around 2.5 years since we started.
Just an add-on to that, some of that was we were going to fly our unit to space frozen, and we had a couple launches that would not have a freezer of the proper temperatures. So that wasn't all just us taking that long, but we used the time when we got it.
Any other questions in the room?
Hi, my name is Maria. I was wondering what inspired you to choose your project?
So I have a career path that I want to share with you. Actually, I kind of wanted to be an engineer in the U.S. Army. So and then this project really inspired me to do that sort of career path. So then I just wanted to learn more about such things, and it really helped me out as a life lesson.
I'm sorry, were you asking how each of the boys chose to be on the team, or were you asking how we chose this particular experiment to run?
Yeah, the particular experiment.
We went to the troop. I'm sorry, I'll answer. You want to go?
Yeah, so we went to the troop. It was a pretty big troop, around like 100 kids in it, and we asked them to submit their ideas. And so we spent a lot of time just filtering through all those ideas. We ended up with about 4 at the end, right? And then we actually combined 2 of those suggestions to get our experiment as it is now.
We started off with 84 suggestions between boys and adults in a night. It took us about 2 more nights to filter through those. We did some rather tricky, get through them quick filtering, and this is what came out of it.
We do have a question from social media.
Yeah, this question asks, what is the suspected cancer-causing substance you're going to use for the experiment?
4-nitroquinoline oxide.
Okay, does that help? We were fortunate enough to have a Abbott microbiologist on the team, and so we started off with salmonella, and then we realized that any scout that got sick in the next 6 months, it was going to be our fault. So we switched to the E. coli, and that brought us back to this particular substance.
Any other questions in the room? Okay, thank you, gentlemen. Thank you to everyone who joined us for today's What's on Board. If you're following along online or in the room, thank you. We're excited to get ready for tomorrow's targeted launch at 12:31 p.m. Eastern Daylight Time. And don't forget, 9:33 this evening, if you're here on Florida Space Coast, look up for the station to go overhead. That's it for What's on Board.